Okay, so today I'm going to talk about some fish that live in seagrass, basically in some really pristine, beautiful habitats, virtually pretty untouched really, and contrary to everybody else, there are very nice, beautiful places in temperate parts and cold parts of the world. Everyone else seems to be in the tropics. So I want to start by talking about deep seagrass habitats. And the deepest seagrass that's been recorded has been down to about 70 metres. It was around 70 metres. But basically nearly all the research we do is in intertidal bays and inlets down to the first couple of metres, probably with the exception of Posidonia oceanica in the Mediterranean, where there's been a lot of research sort of down to 10, 15 metres. But generally we look to those shallow habitats, basically because it's much easier to work in. But as we know, we talked about today, there's more talk about the Great Barrier Reef, but there's estimated to be 31,000 square kilometres of seagrass in greater than 15 metres of water in the Great Barrier Reef area. And although there's been quite a few studies come out in the last couple of years, so they're a bit difficult to read, but there was a fish one in the Chagos archipelago down to about 29 metres. There was a blue carbon one a couple of years ago down to about 20 metres. Generally, they're very, very poorly studied. And not only do we a lot of the times we don't even know what species are there, let alone what ecosystem services they provide. In temperate Australia, there's a lot of seagrass that generally tends to be coastal, and it's often more than 10 metres down to 15, 20, 25 metres. But basically, if you do a search, a literature search, you might find a couple of papers in Western Australia, but otherwise there's very few papers that look at any seagrass greater than 10 metres in depth. But if there's been a few mapping reports, and this is one here. This is out of a report for Tasmania, so down the south of Australia. And this little area here, so this is this section here. Each of these blue areas is just one section that they mapped of the coast. And you can see this figure here actually represents the percentage of Posidonia australis and Amphibolis oceanica, Antarctica, sorry, from one little section. So it's this second section here. And you can see that there's a really, really high coverage of Posidonia in the 15 to 20 metres. But there's very little research done there at all. And actually I was very interested to see in Adriana's talk about she had a site in Tasmania, so that's good that there's actually a little bit of work being done in Tassie. And in this report, they estimated there was about 250 hectares just in this stretch, so about 100 k's, 250 hectares of seagrass greater than 15 metres. So there's a lot of seagrass there, but we almost ignore it, I think. We don't do much. And so at the start of last year, I moved to the University of Newcastle, and every year they do a trip, well, for about the last 10 years, down to Tasmania on this research boat called the Bluefin. It's an Australian Marine College training boat. It's also used for research. And it leaves out of Launceston in the north here, and generally it's used for collection of sponges that are used for cancer research, for some trawling studies, all kinds of different stuff. But because I was going on it, I said I want to do some seagrass stuff. So what can we do in the week that we've got down there? And so a pretty easy thing to do, or easy-ish, is to do fish assemblages in deep seagrass. I thought that's a great opportunity. We don't very often get down there. Not very much research has been done there at all. So I thought, yep, this is perfect. We'll do some baited underwater videos, so some BRUVS. We already had some frames that were down there, so we just extended them a little bit. We had some GoPros. We put some bait on here. So it's a non-destructive measure of sampling seagrass. We used MaxN, which is we had an hour of video for each drop. It's basically, MaxN is the measure you use. It's the maximum number of fish you see in any one given shot. It means that you can't measure one fish more than twice, but there are a few issues which I'll talk about later. And so we ended up in Flinders Island. So this is this island off the north coast of Tasmania. the captain said, I know where there's a heap of seagrass, and so we ended up there, and it's beautiful. It was absolutely beautiful. We stayed there for 2 nights, I think. We were moored up. Only 800 people live on all those islands up there, and basically it's all national park, a little bit of agriculture and some fishermen. So it's really pristine habitats. And if we... How do we get that to go? Sorry. There we go. And this is just some of the Amphibolis beds that were off there. So it's really pristine, really clear water, and really nice seagrass. So one of the other things we're actually able to do is from our camera drops is that we could sort of look at the habitat types as well. The different seagrasses were there and where they were. And we had 25 drops. We had 11 that landed in, and when you drop them, most of the time you can't tell what seagrass is underneath. We had a little bit of idea in the shallow areas, but in the deeper areas we didn't have much idea. So we had 11 in Amphibolis, 8 were in Posidonia, 1 landed in Heterozostera, 1 was in a mixed bed, and we had 1 that actually in about 15 metres that ended in some rubble. So we included that in all of it. We also had 4 video fails, so the ones that landed upside down, and that tended to be unfortunately in the deeper habitats. On the way down they just got tipped up and didn't work so well. So we could still look at the habitat that was there because we could see what seagrass it was. We couldn't actually watch any fish, unfortunately. And what became pretty clear when I started watching these videos pretty early on was that there was a difference in the seagrass species at the different depths. So if you look at this map, we've got the blue is the shallower areas, the green's the deeper areas. So our deepest drop was about 16 metres. So the squares are Amphibolis, and the circles are Posidonia. And here you can see the shallower ones, all Amphibolis, Posidonia deeper. And we zoom in on this one. Posidonia is over here, deeper. There's a couple of Posidonias here, but generally the Amphibolis is shallow and Posidonia is deeper. And so this, I guess if we're going to look at, we can look at the differences in fish between the depths, but it's also going to be related to habitat a little bit. So what did we see? We saw plenty of fish. This brownie coloured fish here was the most common fish. It's the blue throat wrasse. It's a labrinid. That's the female. They were in about 75% of or 73% of our samples. So there was quite a few of them. This is a senator wrasse down here. We saw a few of those, and this blue one is a monocanthid, so a horseshoe leatherjacket. It was our 3rd most common fish. This is a smooth leatherjacket here, a rough leatherjacket. It was our 2nd most common fish in about 55% of our drops. So this becomes interesting because this is the male blue throat wrasse. So the brown one we saw before, this is the male. So they're sexually dimorphic. So one of the issues with Max N was sometimes you'd see a male go past, then you'd see a female go past. Clearly 2 different fish, but by Max N's rules, it's only 1 fish. So it's a very, very conservative measure. We also saw some sharks. So this is a cat shark, a Gulf cat shark. And you can see the water's just crystal clear, and in a couple of the next ones, it's just the fish. So we saw commercial species as well. This is a flathead. So we saw a few of these. And what's really interesting with this one is just how clear and just how nice the seagrass is. It was just beautiful. Another commercially important species, the King George whiting. And these became quite interesting because I only actually saw one King George whiting, and there was a couple of species that were only seen in 1 or 2 shots, and that sort of differentiated our groupings when we look at the MDS plot in a minute. We also saw some sharks. So this is a Port Jackson shark. And the next one. This is in the rubble. So we saw some cryptic species. So this is a weedy sea dragon. So it was quite cool when we saw that. So even though it was only in the rubble, it wasn't in the seagrass, we still counted it. Or didn't still count it. It was still important. And then we saw some rays, we saw a couple of snork, and there's also a squid species that we saw. So we saw a really broad variety of fish and sharks and rays in a whole lot of different habitats. So it was quite cool. And just to recap, yeah, 21 fish species and 1 squid. Not only we also saw some cryptic species. So can anyone see a fish in here? So there's this is a long-tail weedfish, weed whiting. And again here we've got some bridled leatherjackets up here. So they're quite small, but we're able to get them as well. So this is the MDS plot that we did. So just looking at the fish communities in each plot. And what we can see quite clearly here is that these shallow Amphibolis beds grouped up pretty well. There's a couple over here, but they grouped up quite nicely here. Whereas the deeper Posidonia beds were quite different and they're spread out all over the place. So that's kind of the main part. All right, this is my own session, so I'll get moving. If we looked at depth and seagrass species, so the number of species compared to depth, we actually didn't see much difference. It was nearly a straight flat line. So my initial story is not that exciting, but it was still quite enjoyable. I'll cut that one out. And so I guess just to conclusions, these habitats are out there and they provide a habitat for a lot of different fish, both in the shallow and the deep areas. And we probably need to do more research in there. We've got another trip, hopefully if funding comes through, to go again this year. So hopefully we can do some other sampling down there. And sort of as, again, what Adriana was saying before, it's a bit of a hotspot for global warm water warming coming down from the east coast. So it's going to be interesting. It might even just provide a bit of a baseline. So I think there could be time for one question. Can we just let that run? Well, yep. Emma, just quickly.
So your battery life on these was fairly short, and was there a bit of a gap before fish actually started coming into the camera?
So, yeah, usually about 5 minutes, so it wasn't too bad. We got an hour, generally, we got an hour over an hour. So, yeah, so I think you could probably go for a shorter time anyway. Probably half an hour is probably enough anyway. But yeah, so, yeah. In the order of, I know you've got one, Teresa, but well, I can talk to you later. So our next speaker.